Hello, everyone. So I'm Marta Guasca. I'm Associate Professor and Group Leader at CBMR, and it's my great pleasure to introduce our next speaker, Professor Antje Körner, who is a pediatrician scientist, medical doctor, professor of metabolic research at Leipzig University, and she's the head of the Childhood Obesity and Metabolic Research at Health Munich. And her research interest ranges from childhood obesity, trying to understand the underlying mechanisms with a focus on adipose tissue and genetics. Her team has established several well-known cohorts for childhood, such as the Life Child, I know that one. Yeah. And she has earned several prizes, including the Obesity Talent Prize from the ESO and NNF. So we are looking very much forward to your talk on adipogenic hits for kids.
Yeah.
Thank you.
So thank you very much for that very kind introduction, and particularly thank you, Jolene and Kay Wersi and Ruth for inviting me, and also for having that topic on the agenda, because that's a topic that's very dear to my heart. It would be cool if I could see my slides, not my notes. So, you know, I start off the talk with these boring, the boring way with those kind of maps that show prevalences. We have seen that today also on the muscle and I think on longevity. But this map here is on obesity prevalence. So the darker it is, the higher the prevalence, but it's on obesity prevalence in children. And this is a topic that we need to take care about. And I think this is even more visualized by this cool paper, the Lancet paper recently. They did a very kind of simple approach. They looked into the prevalence of disorders in children related to overweight versus underweight. On a global level, as you can see, you can pick your favorite country or area in the world, and you can see in the 90s, of course, there were, particularly in some parts of the world, there was really a lot of people suffering from stunting. But this has dramatically changed. And actually, the UNICEF, I think it was last year, announced that for the first time in humankind, the number of children living with overweight is higher than the number of children living with underweight. So those blue bars in the middle indicate underweight or stunting, and the red bars are indicating overweight. And why I'm so grateful that you choose this topic to be on the agenda is that I think that with all that clinical and scientific advance that we have, there's really a lack for the kids to benefit from this advance. And I think we need to overcome this, and we have also at EASO positioned us in a framework that we need to have this on the agenda and a focus on that. Also, I think that a complex disease like obesity needs a complex approach, and this is what we believe on, that we need a kind of holistic approach to childhood obesity. So, of course, we want to understand what drives obesity in children, as well as the ensuing complications like diabetes, and then how can we detect those high-risk constellation with regard to obesity or also with the driving factors. And of course, we need to understand the mechanisms, and our favorite playground is or has been the adipose tissue, also the genetic, functional genetics, so that we can overcome that prevention and treatment gap. And I also a strong believer that for that we need to connect the experimental and the clinical and epidemiological science. So you've already alluded to that we are employing some cohorts in Leipzig that we have. Of course, I know I'm in Scandinavia, so you have the most beautiful cohorts in the world. But we also have some kind of unique cohorts in Leipzig. Of course, if you want to study metabolism and obesity in kids, you need a cohort for that. So that we do have. Then we do have the Life Child cohort. So this is a populational cohort with recruiting from actually pregnancy into adolescence. And for each of these ages, also have a longitudinal follow-up in these children. And they come to our clinical trial unit every year. And you can see that we also have a lot of biosamples and omics. We have a question registry, and I show you some part of the adipose tissue cohort later on. So, and we are employing this to trying to answer questions. It may be simple questions like, when does obesity manifest? So, as I said, a simple question, and it's a question where everybody has an opinion on it. But of course, to answer it, you need data. You need good data. Now, for those who may not be pediatricians here in the room, in children, obesity is defined based on the BMI, but you use percentiles or SDS. So the cutoff for obesity, at least in Germany, is BMI above the 97th centile. And what we did here is to look into those centile. It's a statistic populational measure over a period of 20 years. And I think you can appreciate two things in this graph. The first is that whereas like the third percentile is pretty stable over the year, the 97th centile is really rising. So that means that not only the prevalence, but also the extent of obesity in children is increasing. And the other thing is that appears to occur very early in life. So, but to answer whether there is something like a critical, a vulnerable age for manifestation of obesity, you need longitudinal data. And for that, we employ data of our QuestNet registry. And what we did is we looked into changes in BMI SDS, so changes in percentile in 1 year interval. So I call it the BMI velocity. And here there's the visualization of the comparison between the normal weight children and those adolescents who have obesity. So every dot above this line of 0 indicates increase in weight in BMI, so increase in the extent of obesity. And you can appreciate that it is actually very early childhood, so between 3 and 6 years of age, where there is the manifestation of obesity. But even after that, it stays positive, so it's getting worse year by year. So, and this is not innocent puppy fat, like we call it in Germany. We also looked into what is the risk that it will stay into adolescence and into adulthood. And here you can see on this graph, what is your chance of ending up with normal weight or overweight when you have overweight as a child with 2 or 3 years, etc. And you can see in infancy, the first year of life, so the chances are 50-50. But already by 3 years of age, the chances or the risk is higher than 90% that the obesity will persist into adulthood. And for those, yesterday we talked with Torben, for those who may be interested also in some endocrinology, if you deal with these kids with obesity, they are really large. So they not only have obesity, they are usually pretty tall too, at least during early childhood. And this is also something that we can appreciate if we compare that on an epidemiological level. So peaking at the age of about 6 years, there's one SDS, one standard deviation score, taller than their lean peers. So this is really a lot. But of course, we're not too much concerned about the height. What we are concerned of is, of course, the complications of obesity. And there are more than 200 complications. We all know them. What we are not as aware of is that these complications also start to emerge early. So, for example, about 30% of kids with obesity already have hypertensive blood pressure value. They have a higher psychosocial and depression burden. And also, and this is what we are really interested in focusing on, is they have a high metabolic risk too. And that implies the next question, which is how can we detect those kids at highest risk for developing early progressive metabolic phenotypes. So from that crowd of kids with obesity, more or less severe, we need to dissect those metabolic trajectories and look into those. It's not a obesity is a heterogeneous disease, so we need to identify those subphenotypes, and particularly those with an early progression. Now, what do we have available, for example, to detect children with diabetes or prediabetes? I mean, we do have those ADA criteria, which is a fasting blood glucose, it's HbA1c, it's 2-hour glucose. And it's always one cutoff level, which means that it is the same for me, as well as for Jolene, for our kids, for our parents, no matter whether we have any condition or not. So wouldn't that require that those metabolic parameters are stable throughout the age? And this is what we looked again at in our cohorts, at very simple measures, as you can see, fasting blood glucose, fasting insulin, which is not one of the ADA criteria, HOMA, IR, etc. And there you can see that there's a tremendous dynamic in those metabolic parameters across the age range. This is true, particularly true for early childhood, but it's also true after childhood. And if you look at fasting blood glucose, you can see that at the old age, the median even crosses the ADA cutoff of impaired fasting glucose. So we do have the clear age dependency, and if we think that further, shouldn't we think about redefining those cutoffs? We are discussing a lot about personalized medicine, how we can be more sharp with that, but we're not discussing how we can really improve the readouts. So seeing this age dependency in these metabolic parameters, we developed, based on a normal healthy weight population, age-specific cutoffs, and we did this also for insulin. And when we applied those cutoffs to a population of kids with obesity, we saw that in very early age, so these are, well, in Germany, kindergarten age kids, about 40% of them already have impaired or hyperinsulinemia, if you like. And on a longitudinal level, we could also show that those age-based and insulin-based cutoffs are more precise and predictive with regard to ensuing dysglycemia later on. And then if you take that even further and look into OGTT, so our glucose tolerance test derived levels, oh, you don't see it, then it becomes even more evident. So if you in addition have not only insulin resistance as defined by Matsuda, but also impaired glucose tolerance test, you have a 12-fold increased ratio for developing prediabetes or diabetes. And this led us to look more deeply into the OGTT-based parameters. And again, we saw that it's particularly the insulin response. So if you have a high and prolonged insulin response in an OGTT, so this confers the highest risk for developing dysglycemia within actually a few years. And what we are doing now, also with the help of, there was a call by Novo Nordisk where you could apply for investigator-initiated trials, and we have been successful. And what we now want to do is to employ those new risk definition and prove that if we start early with the treatment, that we may prevent the manifestation of diabetes and reverse the insulin resistance. So that was for that more metabolic epidemiological part. As I said, our favorite playground is the adipose tissue. And there is this beautiful work from adult medicine. We know that adipose tissue is not adipose tissue. If you have an expansion of a tissue, it can occur via two mechanisms. Either you get more adipocytes, hyperplasia, or you get bigger adipocytes, which is then the hypertrophy. And associated with this, you have a disbalance in the adipokine profile. And I think there's nothing bad with fat as long as we have a healthy, fresh, metabolically active adipose tissue. The problem occurs when we do develop hypertrophy, inflammation, and a disbalance in the adipokine pattern. And this is what is called adipose tissue dysfunction that is associated with insulin resistance. So this is what we know from the adults, but seeing that early emergence of obesity in kids, we were asking how early does this develop? And for this, we employed our Leipzig adipose tissue childhood cohort. So we have samples of more than 700 samples, and depending on the volume of the samples, we do a biological and metabolic and cellular workup in these samples. And one of the first questions that we asked, as I said, was how early does adipose tissue dysfunction develop in kids with obesity? And we could appreciate that there is a hyperplasia in kids with obesity compared to normal weight kids. It's always the normal weight kids are those white ones, and the kids with obesity are the black ones. And as early as we got enough tissue in the samples that we could do the analysis, we found that there is already hypertrophy, so bigger adipocytes in these young children. And with these hypertrophy in the adipose tissue, we could also see that there's a disbalance in the adipokine pattern, like leptin here shown, or adiponectin. And it also correlated with infiltration with macrophages and the formation of those crown-like structures, which we saw in more than half of the kids with obesity and less than 10% in lean kids. And then again, these biological tissue parameters then again were related to proxies of insulin resistance like HOMA-IR. So the essence is that adipose tissue dysfunction develops also very early in life. And what we and many others are now interested in, what are the drivers of this adipose tissue dysfunction, and maybe can we find some targets to revert this adipose tissue dysfunction? So first we started out to look whether there is this kind of clusters in adipose tissue dysfunction in children. So based on those biological and circulating markers, and we could appreciate that there were actually 5 clusters. And interestingly, there were 2 clusters which were more like healthy adipose tissue function, which is the green and the yellow one, and one was younger than, the kids were younger than the other. And there were 3 clusters of adipose tissue dysfunction. One, the cluster C, I think, was more of a secretory type, and the cluster D more of an inflammatory type. And not surprisingly, these were then, of course, also associated with increasing severity of obesity and also with increasing severity of insulin resistance. And what we're going on now is to look whether we can find markers or biomarkers so that we can define those clusters more closely, and compared to those healthy adipose tissue dysfunction, the A and the B cluster, we found 6 genes that were in all of those adipose tissue dysfunctional clusters actually down-regulated. And these are all genes that have never been implicated in adipose tissue biology or obesity or adipose tissue function. Another example, more on the hypothesis-free level, is that what we're doing together with Ellie Zaghini and her postdoc Mauro Tutino is that we wanted to have a molecular landscape of the adipose tissue in children and looking whether there are differences compared to the adults. So what we did a kind of multi-omics approach in our adipose tissue samples. So we did the GWAS, the APIC, and also O-link studies, and we were looking into molecular QTLs. And then in the first approach, we did the molecular EQTLs with existing GWAS data and GTEx data. And in the adults, or compared to the adults, we found much more regulated co-localized genes in kids as compared to adults. And when we looked into what kind of pathways are represented, these were actually pathways involved in adipogenesis and aging. And Ravios asked us to go more deeply into this and look into genes that are regulated also more precisely with age, so with pre-puberty and post-puberty, and we could identify more than 600 genes that do follow these patterns. And another piece, just a glimpse what we are now heading into, is together with the HIMEC people in Leipzig with Lucas Massier and Matthias Bleuer, based on a collaboration with an Israel group in Beersheba. We looked into human adipocyte atlases and trying to identify different subclasses of adipocytes, which we could, again, in the adult samples. And right now, again, we are looking into, is it particularly the same picture that we see in the kids? And again, not surprisingly, we don't. There appear really cellular differences or differences in the prevalence of cell types pre-pubertal and post-pubertal, and also if we specifically map adults versus the kids. And to close up this part, of course, in the end, we would like to go back to what we see clinically, how I started off. And you may remember those pictures. I showed that obesity manifests early. It's also associated with increased linear height in the children. And from a collaboration with Andrew Pospisilek, now in Grand Rapids, he has done an epigenetic profiling based on a bimodal body mass distribution in mice, and he identified a network of genes driven by TRIM28 and the downstream target neonatin. But what I would like to point out that if you look at those mice, they not only have obesity, so they are also taller, so kind of resembling what we see in the kids. Now, what he found was, what was the reason for this, that they really had high insulin levels. So those giant mice had high insulin levels, and he could show that actually they had a beta cell hyperplasia, and he also did all the functional workup, so knocked it down with streptozotocin, and he could reverse the phenotype. And if I now remind you that we also see a very, very early hyperinsulinemia in the kids, that may not be only a consequence, but maybe at some part it may also drive this phenotype. And together with Andrew, we looked into whether there's any molecular signatures in the adipose tissue of the kids as well as the mice. And there you can, by those molecular profile, distinct those gigantic phenotype, and again, it's also in the kids based on the signature associated with hyperinsulinemia. So that's an example how you may bring it all together to some extent. Now, of course, to know what drives obesity, there are probably even in this room, many different opinions. And we may say, well, it's the adipose tissue, and then others may say it's eating habit, and Ruth would say maybe it's all genetics. But of course, we know that it's all of it, and it's our biological makeup that we are always defend our body weight that hits our environment, and the brain is integrating all this. Now, to maybe try to define a little bit more closely what are, let's call it, the risk factors for childhood obesity, again, epidemiology is very helpful. So this is not our data, it's data from the German KICS study in more than 17,000 kids. And here the risk factors, they are ordered by their effects. So the number one risk factor is, of course, if the parents, particularly both parents, have overweight. Number two is a low socioeconomic status. And then you see a lot of perinatal factors, and those classical ones like eating and sleep, etc., is further down the list. And of course, the question is, so what's behind it? Is it the genetics? Is it environment? A shared lifestyle? Is it fetal programming? And to kind of dissect this, I think we need to go deeper into those associations, and if lucky, also try to find some mechanisms. So I've shown you number one risk factor is if the parents have overweight. And not surprisingly, it's shown many times that if the mom has overweight, already by the age of 5 and even before, there's a higher risk for the kids to have overweight and have a higher BMI. So you may think, yeah, okay, it's nature. Well, then if you combine that with environmental or health behavior factors like screen time, then you can see that independent of the weight of the mom, there's an exponential increase in the obesity rate in the kids. So there's an interaction. But it's not only the moms, it's also the dads. So Raffaele Tabarino, he kind of vanished here on the he asked us to look in our live child cohort, what is the contribution of the dad to the obesity risk and the metabolic risk of the kids? So we stratified for the weight ratio, for the weight status of the mom, and no matter whether the mom was normal weight or had overweight, you can see that if the dad had overweight, there was an increased risk for obesity in the kids. Of course, it was the highest if both parents were suffering from overweight. And it was also true for impaired insulin sensitivity. And the nice thing is, why Raf asked us, he had developed a mouse model where he fed the becoming dads or the becoming fathers in the mice a high-fat diet and induced obesity, and then looked into the metabolic parameters. And we looked into commonalities in the transcription profiles in the adipose tissue. And the really cool thing is that Raffaele could really knock down a mechanism for this, which is transmitted via the sperm, via tRNA, that confer this independent contribution of the dad for the offspring metabolic and obesity risk in the children, in mice, as well as in humans. Now, when you're born, there was another conundrum. Many of you may be aware of the Barker hypothesis, which says if you are born small for gestational age, so a small baby, then you have a higher risk for obesity, cardiovascular risk, etc., which is true for cardiovascular metabolic risk. It's not true for obesity. So for obesity, you have a higher risk if you're a large baby. That doubles the risk for developing obesity. And then I think what we need to do is to look into the factors, what makes you a large for gestational age baby. Of course, the first thing you would look at is the mom. And again, we can see if the mom has obesity, that alone doubles the risk of having a large for gestational age baby. And then with every kilogram of weight gain during pregnancy, and that's really starting from the first kilogram, there's an exponential increase in the chance of having a baby with a high birth weight. On the other side, if you're small for gestational age born and then have a rapid catch-up growth, that actually does infer a metabolic risk. This is something that we as pediatric endocrinologists are aware of and did know. And here and another thing is how you could achieve also a catch-up growth is by growth hormone treatment, which also can have a metabolic risk. So we looked into those patient groups, so in normal healthy children, in children who are born small for gestational age, which are the purple one, then children with growth hormone deficiency, so these are usually small and short but lean, and children with obesity. And you can see that before, during, and even after the cessation of growth hormone treatment, that those SGA children do have an increased metabolic risk, and and they also have a a prevalence of dysglycemia, of prediabetes, that's actually not so much different from those children with obesity. So this rapid catch-up does indeed imply a metabolic risk. And after you've born, the first steps into nutrition, and well, here are the experts in this field. Of course, we here know that there's not only the white adipose tissue, but also the brown adipose tissue, and it's good to have a lot of brown adipose tissue because thereby you can burn energy. And when I was in med school, we learned that there is brown adipose tissue just in infancy around your kidneys, and research has shown that there are those scattered brown, beige, white adipocytes within the white adipose tissue. And we used our adipose tissue cohort to look into whether we find those in children as well, which we do, but there was a clear decrease with the degree of obesity and the well-known decrease with the development with age. And here you can see those brown or beige adipocytes stained by UCP1, the marker gene, or you can also see it by the expression. Now, what was interesting when we had been approached by Tamas Röser in Ulm at that time, and said, well, he found again an interesting finding in the mice that that if the mice get mother breast milk, that they could retain this brown or beige adipocytes longer. And here again asked us to whether we could look in our cohort. And that was indeed actually the case. So in those kids who had received breast milk or were breastfed, they were able to longer retain those brown beige adipocytes than those who were formula fed. And we looked in the adipose tissue and also by expression levels. And he did all the functional work about that. But what was really nice for me as a pediatrician, really we pray to the moms, breastfeeding is the best you can do for your child. And it's really cool to see a little piece of evidence that this is actually true. And one quick example about the environment. Of course, what we would need to do is experiments to see whether there is a causal effect, which we cannot do in the children, but the Mother Nature had done it for us in one occasion, and that was the pandemic lockdown with the COVID. And using our Crescent registry, we looked into the prevalence of acute high weight gain in children, and you can see over the last 14 years there was kind of stable, but then it had really risen in 2020, and this was particularly true for those kids who already had obesity. And now we are trying to see whether there's any genetic predisposition that predisposes you to this kind of rapid weight gain. And so we, using our live child cohort, where we had those kids already before the lockdown and then after the lockdown, and we had biosamples before and after. So we did the GWAS and the EPIC chip and the OLIC panel before and after, and we stratified those who were stable in weight, those who gained weight, and those who lost weight. And we're looking into the molecular profiles at those time points. So to start with the genetic predisposition, we employed these polygenic risk scores from adults, admittedly the one published by Kira and Ruth's polygenic risk score, and reassuringly we could see that we could detect the status of obesity or normal weight aligned with those polygenic risk scores. However, disappointingly, when we looked into whether this polygenic risk could kind of predict or whether they have a higher odds ratio for weight gain during the pandemic, you can see that this was not the case. And also if we looked at it as a quantitative trait and looking into the BMR, at the PGSZ scores, we could not see that there were differences in those groups who were stable or who gained weight. So if that is confirmed, it would turn out that the genetic predisposition is not as effective, at least in this admittedly very hard experiment. Similarly, with the epigenetic signatures, there was not a significant association with the groups before or after the lockdown, or with a change in BMI, and also proteomic signatures could not predict the weight groups. But what we could see is that after the lockdown, so at the end, we could see differences in about 37 proteins aligned with those groups. And if you look at those proteins, there are some usual suspects like the leptin, ghrelin is their growth hormone receptor. So, but so far, if you sum this up, it's not so much of a molecular signature that really kind of predicts these weight trajectories in such a severe environmental event like the lockdown. And for the last 4 or 5 minutes, I would like to show you one last piece relating to the genetic background of obesity. We are all aware that there is a genetic predisposition, and of course, for that we need to think in terms of a polygenic predisposition. There are some genes, or most of the genes are overlapping, although there are also different genes. And again, I think we need to understand what do those genes do on a functional level. We were interested, for example, in the functional genetics of TMM18 in adipose tissue. I will not go into the epigenetic stuff, but I want to show you is an example of monogenic background of obesity. Most or almost all of those genes that are implicated in conferring a risk or causing monogenic obesity, they are implicated in the hypothalamic energy regulating pathways. So starting with leptin, actin at the hypothalamus, leptin receptor, POMC, etc., and it all converges down to the MC4 receptor pathway. And of course, it still is a rare disease. So it's less than 5% of patients with obesity suffering from monogenic obesity. But it is important to detect those patients because now we have really treatment options for these patients. And we could recently add one additional of those genes to that list. This was a patient that I saw I think first at the age of 2 with severe obesity. As you can see here, her BMI curve, and actually the gray stuff is the normal, what you expect, the normal range of BMI at that age. And at the age of 12, she was 160 kilogram and more than 100 centimeters tall. And she was also tall for obesity. So the obesity percentile curve here is those obesity-specific normal ranges. So even for kids with obesity, she was particularly tall, and not surprisingly, she had insulin resistance. So this does smell like monogenic obesity, and we did an whole exome, and the panel came back normal. But then she underwent bariatric surgery, and this was when we got an adipose tissue sample of the girl, and we found that her adipocytes were longer able to differentiate into mature adipocytes, and we were looking for the molecular reason for that. And on this volcano plot, one gene really popped out, which is ASIP. Now, what is ASIP? For those of you who may not know, the ASIP is agouti signaling protein. It's usually or normally expressed in the skin, where it acts at the MC1 receptor and regulates the pigmentation of the hair and the skin. And it's an homologue to the mouse agouti. And if you remember, the agouti mice are the oldest obesity mouse model that we know in the world. And this is caused by an ectopic expression of agouti, so that agouti can act at the MC4 receptor in the hypothalamus. And then the phenotype of these mice is that they have kind of yellow, or however you call it, fur, obesity, insulin resistance, etc. So this girl, who is kind of a phenocopy to this agouti mouse, had ectopic expression of ACEP, and we could identify the genetic background. And this is probably the reason why it has not been found before, because it's not an exchange of a single nuclear variant, but it's a chromosomal aberration. So the ACEP gene had been cut off and reintroduced into the genome so that it came under the control of the promoter of another gene, which is itch. And itch is a gene that's ubiquitously expressed in the body. So we proved that this promoter can drive the ACEP expression. We also proved the ectopic ACEP expression, well, in all the cells that we could get hold of. So it's peripheral blood leukocytes, of course, adipocytes, fibroblasts. And we did iPSCs and differentiated them also into hypothalamic-like neurons and could also show the ectopic ACEP expression, and could also show that it does antagonize the MC4 receptor. So that's all published, but it has several implications. The first is, of course, that it helps us to understand more on the human physiology. Second is that looking at this mutation, we need to revise our diagnostic algorithm because those patients are not found right now. And of course, we want to develop treatment options. So for the understanding of the pathophysiology, we developed a humanized mouse model. So we used that human mutation of the girl and introduced it into a mouse background. And here you can see those guys, and you can see who are the ones that are wild type and who are the ones that are the transgenic mice. We proved that there's the ectopic expression of ACEP. You can only see the red bars because the blue bars are really at zero. And we also verified that ACEP is expressed in the hypothalamus, as you can see here, and it also co-localizes with the MC4 receptor. So the prerequisites are there. And when we looked at the phenotype, we could also see that it does copy that of the girls. So the mice were really had severe obesity, the males as well as the females. They were also taller, or however you call that in a mouse, or longer. And they had an altered pigmentation of the fur, although they were not as yellow as other agouti mouse models. And with that, we also saw an increased body fat mass, increased lean body mass, and we could also see that in many organs there was an infiltration with fat. Looking at the adipose tissue, we see again that there's hypertrophy, and here's some pictures and more evidence that these mice developed liver steatosis and also had a metabolic phenotype with impaired glucose tolerance. And of course, the question is, how do they become obese? They have an increased food intake, particularly the females, as you can see here. Surprisingly to us, actually, was that there were no changes in the energy expenditure, but what we did observe and confirm was they had a lower locomotor activity. And this was actually also something that a psychologist told me when she was evaluating our index patient, that these patients do not move as much. And it's also known from other patients, like with leptin receptor deficiency patients, they seem to move less than their peers. Now, the most interesting question, of course, is can we find more patients? And so what we did, again, applied our cohort and looked into cohorts from collaborators, and you can see that we had a decent number of kids and also adults and also lean. That's the last one. And we could find now altogether 26 patients with this trait, and none in the normal healthy lean controls. So for a rare genetic trait, this is really a lot, and it would come second after MC4 receptor if it is confirmed. And we went on and characterized those patients. So they had a severe obesity. They are usually tend to be taller. Hyperphagia is rather moderate. But one feature is what was interesting, and that is that these patients rather had a more decreased or a lower energy expenditure or resting metabolic rate. As you can see here, it also proved to be significant in case control studies, and to further confirm that, we used cell models of these patients and looked into mitochondrial function of primary cells as well as iPSCs differentiated into hypothalamic-like neurons. So that may be a last example how we can learn from extreme phenotypes and really can learn something new. But the essence, I think, of all is that we need to think of all those risk factors, not only the predisposition, which is very important, but also the interaction with the environment and how we can develop measures to predict the susceptibility. We need to understand the mechanisms behind, also leading to the complications, that we can identify modifiable targets, windows of opportunity, so that we can finally help those patients. And with that, of course, I want to thank my team, as well as the Life Child team, and most of all, the patients and the families, because without them, we couldn't do all this research. And the lots of collaborators in Leipzig, as well as nationally and internationally. And I thank you for your attention.
Thank you very much, Dr. Körner, for this very nice talk. Now we have time for a couple questions from the public here.
Antje, that's great. I'll ask you a question about, I'm obsessed with MC3R, and ACIP is beautiful because it's not like AGRP, it only antagonizes MC4R. So you would hypothesize that these children should be tall, which they are because they got no impairment, but you'd also hypothesize that they wouldn't have any delay in puberty, which is what, if you have POMC deficiency, you have severe delay in puberty. So are you getting enough of them now going through puberty that you can time their puberty? And is it normal or is it delayed?
I can't give you a conclusive answer to that. It's not delayed, that I can say. And obviously, the patients are fertile. And actually, in the index patient, she had rather a premature puberty. So she had menarche at the age of 11. And we have another one with the premature puberty, not at the pathological level, and the others tend to be normal. But it doesn't look like delayed puberty so far. Sorry.
That's right. No, that's exactly what I would expect. MC3 is not being impacted on. That's exactly what you would hypothesize.
Yeah. Well, we are doing experiments looking into the affinity of the ACIP to all the MC receptors, and it does have also affinity to the MC2 receptor, not to the M3. Yeah.
Thank you very much. I'm getting back to your first epidemiological studies about the panel of risk factors for childhood obesity. My question is whether you, in this panel of risk factors, actually find somebody that predicts whether the child would get into the disturbed metabolic track or not.
This is exactly the important question, and we are really working on that. So really, how can we identify those kids who are, I call them the early progressors, so who really enter a metabolic alterations and insulin resistance very early. So far, we have improved the risk prediction, but I think we need to employ even more parameters so that we are even more precise.
Obviously, the very first question would be whether metabolic complications in the parents we work on creating the track of metabolic disruption in the child.
So when we looked into the predictiveness of these new reference ranges, we controlled for family history of diabetes, etc. Yeah.
We'll get one last question from Herman over there.
Thank you. Very interesting talk over here.
Ah.
Yes, I think it's a very relevant topic, especially in the context of prevention. So it's an obvious topic to do research on. So I'm happy to see that. My question is in connection to, it's very specific because previous literature has shown that lean children going in the trajectory from lean all the way to obesity, they have a highest risk for cardiometabolic conditions. Mm-hmm. And I've seen that your focus was either on obese or lean with the trajectories. Have you tried to expand this in the context of the GWAS you're running?
What precisely do you mean?
The genetic aspect. So to try to understand with genetics.
Yeah, well, we are in the process of doing it, and again, I totally agree. that it's not only that we not only need to look into the weight category, but also in the dynamics and the trajectories of the weight gain. And this is also what we see, for example, in these SGA children. And we also have a collaboration with a cohort in Rwanda, where they also experience a societal change, and where a lot of previously stunted children, if they have a really rapid catch-up, so they are at metabolic risk. So yeah, I completely agree that you can have that metabolic risk even if you don't have even yet developed obesity.
Thank you.
In the interest of time, we will close this session, but you can go to Dr. Körner, and we would like to thank you on behalf of CBMR for being great.